So we're really obviously interested in the enteroendocrine cell, and I just wanted to really put this picture up here to show the shape of the cell, and also to highlight the fact that these enteroendocrine cells, which are obviously pyramidal in shape, they have nice long processes that tend to go into the lumen to sense what's present in the lumen. But they also have many secretory granules that are mostly found on the basal side. So I should point out this is a basal side, this is a luminal side. And these are the secretory granules that contain hormones such as GLP1 and PYY. So we're really interested in this region of the cell. So I'm sure I'm speaking to people who already know this, but I just highlight that there are lots of different types of enteroendocrine cells, and there is a distribution of enteroendocrine cells depending on the GI tissue that we're talking about. So within the stomach, there's G cells, for example, enterocrine cells, lots of different types of cells within the duodenum and jejunum, and the ileum and the colon. And we're really interested in the colon because we really, like I showed you before, that data to show that nutrient-sensing receptors are expressed here, and therefore really interested in L cells, also because of their ability to regulate appetite by GLP1 and PYY. So how do you, this is just a schematic to show how nutrients and their binding to their receptors have interactions with other cell types. So once nutrients bind to their specific G protein coupled receptors, the enteroendocrine cell becomes activated, and what is shown in the fluorescent image is an activated cell. So we used activation markers like phosphorylated ERK to show this activation. So following this, the switching on of the cell, the secretory granules that contain hormones release their contents, and this release allows them to bind to their respective receptors on vagal afferent endings, for example, resulting in excitation, potentiation, inhibition, to name a few. But they also, of course, enter the bloodstream, so I haven't shown that here. So we've previously shown in humans, so I've just used this symbol to show human because I will be showing some mouse data as well. So that previously we've shown that lauric acid stimulation of GPR84 can specifically activate phosphorylated ERK, and this can induce the release of some of the hormones that I'll show below. So the first panel along the top shows the activation marker phosphorylated ERK, and we've used two different concentrations. So hopefully you can see the green are the activated cells, and when we count the number of activated cells per crypt along the concentration gradient, you can see an increase. We also looked at the release of GLP1 and PYY and 5-HT and found that lauric acid has the ability to increase the release of all three. And so I've shown this before, but I wanted to highlight that I've shown data on GPR84, but we're also quite interested in GPR120 because it is expressed quite highly in the colon and the rectum, so suggesting that it may be a good target for us to look at as well. So just to highlight that GPR84 is stimulated by lauric acid, and we've used lauric acid in all our studies to activate this channel, this receptor. GPR120, which cannot be activated by alpha-linolenic acid, which is found in high levels, for example, in chia and flaxseed. But in our experiments, we use a synthetic agonist called TUG891. Okay, so I'm sure Fiona is familiar with this, but this is a publication by Ekberg and colleagues. What they showed was some really interesting data in mouse. And they showed that GPR119 and GPR40 actually act synergistically on GLP-1 release when studying release from mouse colonic crypts. So if we look specifically at this bar right at the end, what they show is the synergistic effect in orange compared to GPR40 on its own in the blue and GPR119 on its own. And they propose that this synergism is occurring because there are two intracellular pathways that are being activated separately, and then there's convergence at this level of secretion to promote this secretion of GLP-1. And so we have previously shown that GPR84 and GPR120 are actually co-expressed on human L cells. So when we use a stain, we use GLP-1 to stain for L cells, and you can see this specific cell here, shown in the bottom on the merge image, co-localizes both GPR120 and GPR84, so multiple nutrient receptors are on a single L cell. So our research question, therefore, was, is there convergence or synergism of multiple nutrient signal in human colonic enteroendocrine cells? So in order to do this, we use two separate methods. So we use an Using chamber, although it's a modified Using chamber because we're not looking at permeability. We use the Using chamber because it allows us to stimulate with nutrients, specifically the mucosal side of the tissue, whereas the serosal side is kept nice and happy by Krebs buffer that's perfused. So we do an incubation with nutrients for 20 minutes, and then we use immunohistochemistry to look for cell activation markers, pERK and pChem kinase. We also do release assays on biopsies that we've collected. We use some inhibitors to break down to prevent the breakdown of proteins, incubate for 2 hours, collect the supernatant, and then we use a multiplex assay to quantitate these proteins. So I just wanted to show you the mouse data first, because what was really interesting with the mice is that stimulation of both GPR120 with the TUG891 compound activates both pERK and pChem kinase pathways. So if on the red, the green here shows the pERK and the red shows the pChem kinase, and you can see activation of GPR120 stimulates both these pathways. We also did a combination treatment with TUG891 and lauric acid activation, and we saw that the expression or the activation of cells was enhanced by using this combination in both pERK and pChem kinase. When we looked at human tissue, however, we found that there are specific intracellular pathways that are activated. So when we use lauric acid to activate GPR84, we found that only the pERK kinase pathway was activated. And again, you can see this dose-dependent increase. Whereas GPR120, as activated by TUG891, activates specifically the pChem kinase positive pathway. And so when we use these agonists together to activate GPR84 and GPR120, we see that the combination effect has a superadditive effect on cell activation. So if you look at this top panel here, especially this top graph, lauric acid on its own does increase phosphorylated ERK expression, but this is almost 4 times as much when we use a combination treatment. Phosphorylated chem kinase on its own also does activate pChem kinase, like I showed before, but again, when we use in combination, we almost double the number of activated cells. And perhaps most importantly, this combination treatment induces a synergistic effect on the release of GLP-1 and PYY. So you can see GLP-1 in this graph. Again, lauric acid and TUG does induce release, but we see almost a doubling effect when we use a combination treatment. We've also looked at PYY, and again you see this massive increase in the amount of PYY that is released. And so we conclude that the synergism and amplification we see by nutrient activation is perhaps occurring because we've got lauric acid binding to its receptor GPR84 and activating a pERK kinase pathway, resulting in hormones to be released, binding to their receptor, and entering the bloodstream and having central effects. But together we see TUG891, and when we add TUG891, activating GPR120, we activate another pathway, the pChem kinase positive pathway, and this increases the amount of hormones that is actually released from these cells, and so you get a bigger signal actually occurring into the CNS. So in the future, we'd like to know what are the future research questions is this going to happen in vivo? So can multiple nutrient stimulation of colonic L cells actually be used to modulate appetite in vivo? And we have a clinical trial in progress, so hopefully we should find this out sooner rather than later. I'd also really like to know whether multiple nutrients can actually alter the proliferation and differentiation of cells that we just discussed before, and also whether the receptor phenotype of a cell can be changed by the multiple nutrients that we're testing. And I'd just like to thank our funders and thank everyone for listening to me again, and I open the floor to questions.